You guys like Madonna? You don't have to like Madonna, but you might know her song Material Girl. It goes, we are living in the material world. I am a material girl. I have my own version. It goes, we're living in a bacterial world, and I'm a bacterial, well, boy, but the point is we're living in a bacterial world. Everything from deep ocean vents to the tops of mountains to every surface that we touch is coated in bacteria. Madonna, whether or not she knows it, is a bacterial girl. 10 times the number of cells in and on Madonna are bacterial as opposed to of human origin. This tube of soil that I have right here, there's 1 gram of soil in this tube, and an estimated 10,000 distinct organisms. Upwards of 10,000 distinct species, upwards of 1 billion organisms. These 10,000 species all have genomes. Each one of those genomes has about 5,000 genes in it. That means we have 50 million bacterial genes in this one tube right here of soil. And these genes are like no genes you've seen before. These genes are like no genes anybody has seen before, literally. These genes encode proteins which are about 55% identical to any protein we have in any sequence database. To give you some context, that's about as related as humans are to a banana. Think about all the things humans can do that a banana cannot do. Now think about all the things that might be here in this soil that we've never seen before, things that we don't know can be done. There's a lot of exciting discoveries to be made in this 1 gram of soil. But here's the catch. Only 1% of the bacteria in this gram of soil, only 1% of them can be grown in a lab. So far, we've only studied about 1% of the genes in this soil. These 1% have done a lot. We have antibiotics from the 1% of bacteria that grow in this soil. But the other 99%, we haven't studied microbial dark matter. But we can study that starting today. And the reason we can study that today is because we have tremendous sequencing power. We don't have to grow these organisms. We can sequence the majority of DNA that's in the soil. We can then scan that sequence. We can predict what genes in that sequence are going to give us useful functions. Then we can test for those useful functions. We take those genes, we put them into bacteria that we can grow in the lab. We put them into bacteria that we do understand. And then we test those bacteria for useful functions. This idea of putting genes in a bacteria, it's been around for a long time. That's how we have insulin. Take 2 genes, put it in E. coli, make insulin, save millions of lives. Many drugs, many chemicals, many fuels, many useful things, bioremediation efforts have come from putting genes into bacteria, but we've only, up to today, used those 1% of genes that we do know. We haven't started investigating the other 99%. We're on the edge of a frontier. We can do some really exciting things. It's not going to be easy. It's not going to be easy to figure out what genes are useful, what genes aren't, what genes are neutral. There's going to be good genes, there's going to be bad genes. So we're going to have to be careful. But I'm excited, I'm thrilled, I'm optimistic for what we can find in 1 gram of soil. And I hope you are too. Thank you.